In previous videos, we discussed how sperm are produced, but we didn't actually talk about what sperm are made of. The sperm itself has three main regions. The first region is the head region. The head region contains all the genetic code, contains all the DNA. But surrounding that head region is what's referred to as an acrosome. The acrosome is important because it contains hyaluronidase. Now, hyaluronidase is going to be an enzyme that can digest or break down things that the head comes into contact with. Thinking, well, why would you want to break down and digest? The head has to be able to enter the actual egg. The hyaluronidase helps to digest the outside of the coating of the egg. We then have the midpiece. Midpiece is where most of the mitochondria are located. This is where you have the metabolic activity occurring, because the sperm have to stay alive to get to the egg. And then we have the tail. The tail is the flagella. This is locomotion, movement. It's how the sperm propels itself through the female reproductive system to try and find that egg, that basic gamete. So what we can see here is, you can see the acrosome. That is encasing around the head, encasing around where the genetic material is, which means it's there so if anytime that head bumps into what it wants to, the egg, it can release the hyaluronidase, release the enzyme. Then you go down to the midpiece. You can see that's where the mitochondria are located. And then you have the long flagella at the end, which is the tail. That's for locomotion purposes. All right, sustentacular cells. Now, these don't directly actually make the sperm, but they play an important role. They're there as the sperm cells are being produced. So the original stem cells all the way through meiosis. They're meant to be supporting cells. They're providing nutrients, and they help to signal when those stem cells should divide. So helping to control part of this process. But they're having more than just that as the purpose. They help to secrete the testicular fluid. Now that testicular fluid, while it's absorbed later on in the process, at this point, it helps facilitate the movement of the non-motile sperm. But probably the most important aspect is the tight junctions that form this blood-testes barrier. The sustentacular cells are all tight junctions all the way around the entire structure. This prevents any of the sperm antigens from getting into the bloodstream. They're all contained only in the testes, only where they're being produced. Saying, well, who cares? Why does it make a difference? Because those sperm antigens would activate the immune system. Now, if you're thinking, hold on, your cells have this self-antigen. They shouldn't activate. True. But that self-antigen on the cells, it was there as your immune system was first developing. That was developing as a young child. The actual sperm themselves are not produced until puberty. That means these sperm cells do not have the self-antigen, which means your body is going to destroy them if they're found, because they're produced after the immune system made its listing of self-antigens. So therefore, because the sperm aren't there prior to puberty, the immune system recognizes them as foreign. They don't have that same self-marker because it was never recognized because it was never there when the immune system was forming. So here is that sustentacular cell. They're kind of forming these barriers, these junctions. Here you can see some of the tight junctions are located between the two sustentacular cells. It allows for the developing sperm to move through, but otherwise helps contain everything, keeping the antigens in place, preventing the body's immune system from destroying these cells, which it thinks are foreign, because it can't recognize them. They weren't there at the start. But while we've talked about how these are produced, we haven't talked about any control mechanisms. The control mechanism is the hypothalamic pituitary gonadal axis, which means we're pulling the endocrine system back in here. which makes sense because we've talked about the gonads in the endocrine system. So we'll start off first with GnRH. Hypothalamus is going to release gonadotropin-releasing hormone, GnRH. That gonadotropin-releasing hormone goes from the hypothalamus down to the pituitary gland, specifically the anterior pituitary gland. In the anterior pituitary, we are going to stimulate and release, so produce and release FSH, follicle stimulating hormone, and LH, luteinizing hormone. Those two will go into the bloodstream and they'll be head to foot, but we're focusing on what happens when they reach the testes. You're going to have both of them stimulating different cells. The FSH is going to stimulate ABP. Now, ABP is androgen binding protein. So you need this FSH to stimulate the androgen binding protein because that makes the cells receptive to testosterone. But now the other hormone, looking at LH, is going to stimulate the interstitial cells. Interstitial cells, hopefully remember, are what produce testosterone. So that means LH, luteinizing hormone, is stimulating the production of testosterone. While FSH, follicle stimulating hormone, made the actual cells receptive to testosterone. They triggered that androgen binding protein. So you have to have both of these, FSH and LH, working together. That allows testosterone to then be an activation for the androgen binding protein, which the spermatic cells then become receptive. Once those spermatic cells are receptive to testosterone, and there's adequate amounts of testosterone, then you have the final trigger for spermatogenesis. That's why it doesn't occur until puberty. Because while the other hormones can be present before, testosterone and the amounts of these being produced doesn't really kick in until puberty itself. So it kicks in high enough levels of FSH, LH, testosterone to now trigger, being the final trigger for spermatogenesis. But that's not the only purpose of testosterone. It also heads out to cause other physiological effects throughout the body. It has impacts in muscular development, has impacts in the neural development. There's many other systems in the body that have the impact here. But that's just the start. There's also feedback loops. You're going to have this negative feedback, which is coming from testosterone. Too much testosterone is going to go back up to the anterior pituitary and say, hey, there's enough testosterone, cut down production. Reduce FSH and LH. Also, you're going to have this inhibin, which can be produced by the spermatogenic cells. Inhibin can then also inhibit FSH and LH production. By doing that, you reduce those amounts and therefore reduce spermatogenesis. So two negative feedbacks to help control the amount of production of sperm in the body. So a little about the physiology of how the sperm are controlled, as well as the structural parts of the sperm.